Good afternoon. Welcome to my class. I'm Teacher Irene, your science teacher for today.
Good afternoon, class. Good afternoon.
Good afternoon.
We will have a new lesson for today. But before that, let's have first a prayer. May I call on the president of the class to lead the prayer?
Let's bow our head into the presence of the Lord. Lord, help us in our class today. Give us concentration so that we listen, understand, learn, and with a peaceful mind, we always remember that Jesus Christ is always with us.
Amen. Thank you, Franz. And now let's have the checking of attendance. Angel, can you please check the attendance?
Good afternoon, ma'am. We have 15 students today, 5 boys and 10 girls.
Very good. We have a perfect attendance for today. For our online classroom rules, first, you have to choose a quiet and comfortable place to study. Second, use your headphone to minimize noise distraction and clearly hear the audio. You have to mute your microphone when someone is presenting. Actively participate in the online discussion. And enjoy learning. And now let's have a game. It's time to climb by Nerpad. You have to race to the top of the mountain against the clock by answering the 5 multiple choice questions. This one will unblock the difficulties and recall the previous topic about the comparison between DNA with RNA and DNA replication using the educational game nerpad.com. The first student who climb will be the winner and will get the 10 points in the recitation. So let's go, guys. Okay, so we have 15 players. Let's start. Okay, for the first question, what are the 4 nitrogenous bases of DNA? Leading player, Samonte. Second question, which shows the correct complementary base pairing for DNA? Third question, what are the 4 nitrogenous bases of RNA? Leading player, Shania. For the fourth question, what is the pairing of the nitrogenous bases in RNA? And for the last question, number 5, DNA replication results into DNA molecules. And leading player, Shania. Okay, congratulations, Shania, with a score of 4,961 points. Very good. You will get the 10 points citation. Good job, Nap. So now let's go back to our PowerPoint presentation. Let's check your answer. For the first question, what are the 4 nitrogenous bases of DNA? Anyone who wants? Yes? Name, please.
Pareha po.
Yes, Pareha. What is your answer here? Yes?
Letter C po.
Letter C. Let's check. And letter C is correct. The 4 nitrogenous bases of DNA are adenine, thymine, cytosine, and guanine. Very good. And let's have number 2 question. which shows the correct complementary base pairing for DNA. Volunteer, please. Anyone? Yes? Name, please.
William po.
Yes,
William po.
William.
Letter D po.
Letter D, let's check. Yes, correct. Okay, we have thymine paired with adenine and guanine paired with cytosine. Very good. Let's have number 3. What are the 4 nitrogenous bases of RNA? Anyone? Volunteer?
Ma'am,
Name, please. Yes?
Trisha po.
Yes, Trisha.
Letter C po.
Letter C, let's check. Yes, correct. So the 4 nitrogenous bases of RNA are adenine, uracil, cytosine, and guanine. Here, in DNA, you have the thymine, and in RNA, you have the uracil. And let's have the 4th question. What is the pairing of the nitrogenous bases in RNA? Name this, volunteer, anyone?
Ma'am.
Yes? Name please?
Nathan po.
Yes, Nathan. What is your answer for number 4?
Letter D po.
Letter D, let's check. Yes, it's uracil paired with adenine and guanine paired with cytosine. Very good. Let's have number 5. DNA replication results in 2 DNA molecules. Name? Name please. Any volunteer? Yes?
Sean po.
Sean, yes?
Letter C po.
Letter C, let's check. Very good. It's letter C. Each one with one new strand, that is the gray in color here, and one original, the color blue. Okay, now, I have here, I prepared a puzzle, and you have to crack the code. Your task is you have to decode the secret message that is found on the left side, and your hint is found at the right side. If you know the answer, you have to type it in the chat box, and the first one who will type the correct answer will get the 10 points in the recitation. So let's start. Okay, very good. And the correct answer is protein synthesis, transcription, and translation. And the first one who give the correct answer is Arabella. Okay, very good, Arabella. You will get the 10 points in the recitation. Okay, actually, guys, the puzzle is related to our new lesson for today, which is protein synthesis, where the puzzle where we convert the number to letter to form the word and then to form the sentence. Like in protein synthesis, the DNA is converted to RNA in the process of transcription, and RNA converted to amino acid to form the proteins by the process of translation. Okay? So, let's have again, the title is Protein Synthesis, Transcription, and Translation. And for our learning competency is you have to explain how protein is made using information from DNA. And for our specific objectives, at the end of the lesson, at least 80% of you guys should be able to explain the events of transcription and translation in protein synthesis. Second, demonstrate the process of transcription and translation using the given DNA strand and relate the importance of proteins in the human body. Can you hear me, guys? Hello, good afternoon.
Yes, ma'am.
Okay, let's go back. So again, guys, I prepare here a video to discuss to you what is protein synthesis, what is transcription and translation. Did you see my screen?
Yes, ma'am.
Yes.
Yes, po.
Yes. Okay, so you have to watch the video and after that, I will ask some questions. Okay? Ready?
Yes, po.
Hi! After learning about DNA, have you ever wondered how can the DNA actually result in a trait? Let's take an example like eye color. Yes, your DNA has a genetic information that codes for the color of your eyes. Your eye color is based on the pigment that is inside the eyes. But in order to have that pigment, you have genes, which are portions of DNA that can code for proteins which help make that pigment. So what we're to talk about is about your DNA can lead to the making of a protein. This process is called protein synthesis. Synthesis essentially means make something. So protein synthesis means to make protein. What's the big deal about proteins? Proteins are kind of a big deal. They do all kinds of things. Proteins are involved in transport, in structure, in acting as enzymes that make all kinds of materials, in protecting the body, and so much more. Proteins is essential for you to live. Protein synthesis is the process where a sequence of DNA is used to build a protein from individual amino acids. The two processes or steps in protein synthesis are transcription and translation. It's a transfer of information from DNA to RNA to proteins or gene expression. Let's try to answer this one. Let's complete the diagram and identify the amino acid sequence coded by each mRNA. The given DNA strand are A, T, G, G, T, A, G, C, T, A, C, C, T, and D. First, we have to fill in the complementary strand. Remember, T-thymine pair with A adenine and G-guanine pair with C cytosine. So here, the complementary DNA strand is T, A, C, C, A, T, C, G, A, T, T, G, G, A, and A. The first step in the process is called transcription, where a coding region of DNA is converted to messenger RNA or mRNA, the first type of RNA. Here, it transcribes or copies DNA's message or the genetic code in nucleus. Part of the DNA temporarily unzips and is used as a template to assemble complementary nucleotides into mRNA. During transcription, mRNA is made from the DNA sequence, following the base pair rule in RNA. Uracil pair with adenine and guanine pair with cytosine. For the second procedure, we have to fill in the correct mRNA bases by transcribing the bottom DNA code. So here, the mRNA bases are A, U, G, G, U, A, G, C, U, A, C, C, U, and U. The second step or process in protein synthesis is translation. This is the process of converting or decoding the information in mRNA into a sequence of amino acids to make a protein. In translation, The mRNA leaves the nucleus and gets to a ribosome in the cell cytoplasm. The ribosome reads the message 3 bases at a time. The 3 bases of mRNA is called codon. It will code for one amino acid. The amino acids are joined together and folded into a protein. Transfer RNA or tRNA, the second type of RNA, carries amino acid from the cytoplasm to the ribosome. tRNAs join and bring correct amino acids. Each tRNA has 3 nucleotides that are complementary to the codon in mRNA. The 2 parts of tRNA are anticodon and amino acid attachment site. tRNAs move from A site to P site. Then the A site is now open for new with attached amino acid to join. For the third procedure, we have to fill in the correct tRNA bases. So we have U, A, C, C, A, U, C, G, A, U, U, G, G, A. The third type of RNA is the ribosomal RNA or rRNA. Allows amino acid to attach by making peptide bonds or to assemble protein. The ribosome recognizes a stop codon. The polypeptide is terminated and released. For the fourth procedure, translate the mRNA codons to find the correct amino acids. We will use the genetic code. The genetic code is the language in which instructions for proteins are written in the base sequences. It is used to decode mRNA message. So here, the first letter is A, second letter is U, and the third letter is G, and that is methionine. The G, the U, and the A is valine. The G, C, and U is alanine. A, C asparagine, and the C, U is the leucine. So the amino acids here is methionine, valine, alanine, asparagine, and leucine.
So now guys, let's go back to our PowerPoint presentation. And the first question is, what is protein synthesis? Volunteer anyone in the class?
Mom,
Yes? Name please?
Jason,
Yes, Jaisal, what is your answer?
protein synthesis is a process where DNA is used to build a protein from individual amino acids.
Very good, Jaisal. When we say protein synthesis, this one is a process where the DNA is converted to amino acid to build or form the protein. Okay, for the second question, what are the two processes or the steps in protein synthesis? Anyone volunteer?
Mom,
Yes? Name please?
Jillian po.
Yes? Jillian?
The two steps in protein synthesis are transcription and translation, ma'am.
Very good. That is transcription and translation. And when we say transcription, this is the DNA is converted to mRNA, while translation is the mRNA is converted to amino acid to make a protein. Very good. And now, actually, guys, our lesson for today is related to ESP subject in terms of love, marriage, or relationship pairing of or soulmate. Since once you are in love, enter into a serious relationship, and got married, you are already committed to each other. It means you need to devote your love, faith, and loyalty to your husband and wife only because he is your partner in life forever. And once somebody paired to another, there will be a negative effect to everyone in the family. Just like in pairing of nitrogenous bases in DNA, the thymine with adenine and guanine with cytosine, and in RNA, the uracil with adenine, guanine with cytosine. If these nitrogenous bases paired with different or wrong partner, there will be a negative effect, which is what we call mutation. And this topic, guys, will be discussed next week. Or any reaction, guys, on what I said? Any reaction?
Agree po, agree po.
Okay, very good. And now it's time to check, or it's time to solve this one. Let's practice solving. Okay. So the given DNA strand here is A T G G T A G C T A A C C T T. Okay, so what is the first procedure we need to do to answer this one? Okay, volunteer. Okay, yes.
Shania po.
Yes, Shania.
The first, pros- the first procedure is to fill in the complementary DNA strand po.
Okay, very good. You have to fill in the complementary DNA strand. Very good. So what is the DNA, or what is the complementary DNA strand here? Anyone in the class who wants?
Ma'am,
Yes. Yes. Name?
Angel.
Yes, Angel.
Angel.
Yes, Angel. What is your answer? Check.
TACCAECGATTGGAA.
Very good. Very good. How about the second procedure? What is the second procedure, or the next step we need to do to complete this one, to solve this one? Yes. Name, please.
Nathan po.
Yes, Nathan.
Fill in the correct mRNA bases by transcribing the bottom DNA code.
Okay. Fill in the correct mRNA bases by transcribing the bottom DNA code. This one, the red one. Okay. To get the mRNA. So what is the answer here? Any volunteer from the class? Volunteer?
Ma'am,
Yes. Name, please.
ma'am,
Yes.
heart cell po.
Heart cell. Yes, heart cell.
AUGGUAGCUAACCUU.
Very good. Very good, heart cell. Okay, how about the next step, or the next procedure we need to do? Anyone from the class? Yes?
Trisha po.
Yes, Trisha.
And the third procedure is to fill in the correct RNA bases.
Okay, very good. You have to fill in the correct tRNA bases. Very good. Okay, so what is the tRNA here? Anyone from the class? Yes? Name?
Jason po.
Again?
Jason.
J cell. Yes, J cell.
UACCAUCGAUGGAA.
Very good. Very good, Jaisal. How about the last procedure? What is the last procedure we need to do?
Ma'am,
Yes? Yes, Jillian.
to translate the mRNA codons to find the correct amino acid.
Okay, we have to translate the mRNA codons to find the correct amino acid. So we are using this one, the color brown, to get the amino acid, or from mRNA to get the amino acid. And what are we going, or what we need to use to get the name of the amino acids?
Ma'am,
Volunteer, yes. Name, please.
Arabella po.
Yes, Arabella, what is your answer?
We will use the genetic code to find the correct amino acids.
Very good, very good. Okay, so what is the amino acids here? Anyone in the class? Volunteer?
Ma'am,
Yes, name please.
Chelmar po.
Yes, Chalmar.
AUG is methionine. GUA
Very
is
good.
valine.
Again,
GCU
again, we have GUA is,
valine.
yes, Balian, and
GCU is alanine.
very good,
AAC is asparagine.
very good.
And CUU is leucine.
Very good, Chalmar. Okay, very good. So let's proceed. Again, when we say the first letter here is, we have A, second letter is U, and the third letter is G. So let's check. Again, the first letter is A, second letter is U, and the third letter is G, where the three letters combine or meet. That is the name of the amino acid. And the name here is, we have the methionine. So now it's your turn, guys. Let's divide the class into three. We have three groups, group A, B, and C. You need here a paper and a pen. Can you show please in the camera your paper and pen? Are you ready? Okay, very good. For the group A, we have Fran, Shania, Sean, Gina, and William. For the group B, Angel, Hartzell, Leeward, Nathan, and Joseph. And for the group C, we have Trisha, Arabella, Chalmar, Elaine, and Aliana. Take a screenshot, please, for the group A. This is the given. Take a screenshot. Okay. Let's have the group B. Take a screenshot. Okay, let's have the group C. Okay. So you have 5 minutes, guys, to do the activity. You have to use your group chat to communicate with each other. And remember, honesty and teamwork, or helping with each other and doing your best in answering the activity. Start now. Again, you have to use your group chat to communicate with each other. You have to take a picture and post it in the group chat, guys. Your answer step by step. Angel, very good for the group B. That is the given. Yes, very good. Very good, guys. Let's have the group A. Okay, very good, Franz. You have to fill in the complementary DNA strand. Yes, correct. Very good.
The third procedure is to fill in the correct tRNA bases, and the answer are GB, CC, GUAA, CGA, GCUA.
Very good.
So lastly, translate the mRNA codon to find the correct amino acid. First, we have letter C, second A, and third is G, which is glutamate, and
Check.
G, AA, and which is glutamic acid,
Check.
UUG, which is leucine, CUC, which is leucine, GAU, which is aspartate. And the amino acids are glutamate, glutamic acid, leucine, and aspartate. Thank you.
Very good, group A. And let's have the last group. Let's have the group C.
Let's go, Group C.
Very good, Trish.
Good afternoon, ma'am. Good afternoon, classmates. The given DNA strands are GTA, CTC, AAG, GTC, AAG.
Yes.
The first procedure is fill in the complementary strand. The answer are CAT, GAG, TTC, TAG, ATC.
Check. Very good.
Our next procedure is fill in the correct mRNA bases by transcribing the bottom DNA code, and these are GUACUC, AAG, GUC, AUAG.
Very good.
The next step is to fill in the correct tRNA bases, and
Yes.
the answers are CAU, GAGU, UCC, AGAUC.
Very good.
And lastly, translate the mRNA codons to find the correct amino acid. First letter we have G, second letter is U, third letter is A, which is valine, CUC, which is leucine, AAG, which is lysine, GUC is valine, and UAG, which is stop. And the amino acid here are valine, leucine, lysine, valine, and stop.
Stop or end. Very good, group C. Very good. And now, let's check the application of our topic. Can you please share something on the application of our topic in daily life? Any volunteer from the class?
Ma'am?
Yes.
Any volunteer from the class?
Name?
Let's go.
Yes, Fran?
Yes. Finding the real parent through DNA
Yes,
sample.
Okay, actually, in fact, I know that
Finding the
all of
two
you
parents to make
are,
example.
yes, very good. In fact, I know that all of you are familiar with the Jessica Soho show where KMJS used DNA samples to find the real parents of the child. And actually, did you know that there are two applications of DNA analysis in forensic medicine, that is in criminal investigation, and the second one is in paternity testing. And now, let's summarize what you have learned. You have to answer the following question based on the objectives. You have to type your answer in the booklab.com with the code of BXPFUQ. Let's go, guys. Okay, let's start. The first question is, explain the events of transcription and translation in protein synthesis. Yes, very good. When we say transcription, yes, when mRNA is converted to amino acid, very good, guys. And translation is the process of converting the information in mRNA. Yes, very good. Let's check. Okay, very good. Any volunteer who wants to explain or share her idea or his idea about the question or his answer in question number one? Anyone in the class? Volunteer? Yes?
Heart cell po.
Yes, hard cell?
Protein synthesis. Protein synthesis. DNA is converted to acid to form protein. It occurs in two steps: transcription and translation. Transcription, DNA is converted to mRNA, while translation, mRNA is converted to amino acid to make a protein.
Very good with the complete answer. Okay, let's have the second question. You have to demonstrate the process of transcription and translation using the given DNA strand. You have to type first the different procedure to get the process on how to transcribe and translate using the given DNA strand. Okay, the different steps, yes. Yes. The first one, yes, is fill in the complementary strand, check. Fill in the correct mRNA base by transcribing the bottom DNA code, yes, check. Fill in the correct tRNA base, yes, check. Translate the mRNA codons to find the correct amino acid. And also, we need to use that genetic code. Very good. So, anyone in the class who wants to demonstrate the process of transcribing and translation and transcribing using the given DNA strand in the previous activity. Anyone for the class?
Ma'am,
Yes?
Angel po.
Yes, Angel?
So this is our group. So in our group B, the given is TTT-CAA-TCGA-CCAAC. First procedure is you need to fill in the complementary DNA strand. So the complementary DNA strand is AAA-GTT-AGCT-GGTTG. Second procedure is you need to fill in the mRNA bases by describing the bottom DNA code. So the mRNA code is UUU CAAUCGACCAAC. For the third procedure is fill in the correct tRNA bases. So the answer will be AAA-GUU-AGC-UGG-UUG. For the last step is translate the mRNA codons to find the correct amino acid. We use genetic code to find the correct amino acid. So UUU is phenylalanine, CAA is glutamine, UCG is serine, ACC is threonine, AAA is asparagine.
Very good, Angel. Good job. And now for the last question. What is or are the importance or application of proteins in the human body? Again, what is or are the importance or application of proteins in the human body? You have to type it in the vocab. Very good. Protein is important for growth and development in children. Very good. Yes. Human body contains protein. Very good. You need protein in your diet to help your body repair cells. Very good. Yes, check. Protein is important for growth. Okay, so anyone in the class who wants to share his or her answer for this question? Yes?
Liver po.
Yes, Leeward?
Protein is important for growth and development in children po.
Very good. And actually, protein is found in different parts of our body. In our hair, nails, muscles, cellular messengers, antibodies, blood, brain, and nerves, enzymes, and cellular construction workers. So now, guys, any questions or clarification about our lesson for today?
None po.
None? So if yes, so let's start. It's time to take a quiz. You have to answer the 5-item multiple choice using the Socrative.com with the code of Evangelista6727. So let's go, guys. 15 students. Okay, so for the first question, once transcription has been completed, which of the following is not necessary for protein synthesis to occur? tRNA, ribosome, mRNA, or DNA? 12 students, 13 students answered, 14 students. Last one. Last one. 14 out of 15 students answered. Oh, maybe because of poor connection. Okay, we have 15 out of 15 students answered. Let's have the second question. For the second question, complete the sentence. During translation is blank. DNA is made complementary to RNA. RNA is made complementary to DNA. A protein is made from the RNA-based sequence, or a protein is made from the DNA-based sequence. Okay, 15 out of 15. Let's have number 3 question. What is the basic rule of mRNA? 8, 10, 12. Very good. 14 out of 15. Waiting. Yes, 15 out of 15 students answered. Let's have number 4. If the DNA code reads as a CCAATTGG, then the tRNA code would read as 13 out of 15, 14 out of 15. Okay, very good. 15 out of 15 students answered. Last question for number 5. Some proteins provide structure in our body. For example, blank gives structure to your hair, nails, and skin, while blank can be found in various connective tissues to provide structure inside your body. 13 students. Okay, 14 students. And waiting for the last student to answer. 15 out of 15. Let's check your answer. Show result, responses, names. Oh, very good. Aliana, Angel, Arabella, Chalmar, Gina, France, Parcel, Jaisal, Jelaine, Leeward, 5 points. Nathan, Shania, 4 points. Trisha, 5. William, 5. Sean, 5. Very good. For item number 1, 100% got the correct answer. For number 2, 100%, 93% correct answer. For number 4 and number 5, 100% got the correct answer. Very good, guys. Very good. And now, to understand about our new lesson today, you have to play the simulation on this website. I will send this one in your group chat, okay? So, thank you guys, and see you next week.
I hope you enjoyed
Thank you.
the example today.
Thank you, Mama.
Thank you.
Until
Thank
next
you po,
time.
ma'am. Thank you.
Bye.
Thank you po.